Okay, we just have one more thing to talk about on this slide, and this is this concept of denaturing, denaturization, denaturation. Sorry, try to put a different sh in there. And you experience this when you boil an egg. The egg starts out the yolk, which the yolk and the white, but particularly the white part of the egg starts out translucent. You can see through it somewhat, and as you cook it, it becomes opaque. You can see that change. What's happening there is the proteins are becoming denatured due to heat. High heat will denature protein. It causes the structure to change. It breaks down the hydrogen bonds, and that change in structure renders it inactive. It's not active as a protein anymore. It still works for us. We can eat it, and we eat it, we ingest it, we break it down into amino acids, and we can then use that as building blocks for us because we're building new proteins, but it denatures that protein. This also happens in your body. It's one of the reasons why a very high fever is very dangerous, particularly in people who are actually, a high fever is dangerous for anybody because much of your body is proteins. And so that high fever, that high heat can denature your proteins. You have proteins in your brain. You have those enzymes that are proteins, structural proteins, antibodies. Your heme, your blood, is protein based, and that then could possibly render them inactive, and you need those things very much. That unraveling, the denaturing can happen because of an increased pH, salt concentration, temperature, or other unknown or known environmental factors. If it happens, it's generally not a good thing. Wow, this slide got messed up. But anyway, the slide is about sickle cell anemia, and what you have is a slight change in the primary structure. So sickle cell anemia is a problem with the red blood cells not being able to carry oxygen because their shape has changed. And so you have just one of the amino acid you have a change of just one amino acid in the sequence in the primary structure. That then causes the secondary and tertiary structures to be shaped different. That structure, and particularly the normal structure of the quadrantary, that structure of the hemoglobin is what allows it to carry oxygen, and that structure has been changed in the sickle cell, so no longer a functioning I guess healthy functioning. I mean, the sickle cell can carry oxygen to a certain degree. It's just not as effective. But the other thing is you have this hemoglobin molecule that now have edges that it is not normal, so it can lead to strokes. They can actually get together. The fourth type of macromolecule we need to talk about is a nucleic acid. Nucleic acids are the building blocks of our hereditary information, and so they're the building blocks of our genes, and we study these, or the building blocks of genes of whatever species you're looking at, and we study these because we are looking at the sequencing and looking at what is normal and comparing that to abnormal, and looking for where the abnormalities are to try to address certain incurable diseases. We call the study of a specific species genes genomics. We call the study of the proteins those genes make proteomics, and the discipline that studies this is the bioinformatics. And so you actually have that at the end of this chapter. And I happen to know I didn't really include any slides on that, but I did want you to know that those terms are out there. So amino acid sequence of a polypeptide is programmed by the gene. You have your genes in your nucleus of your cell. They actually dictate what proteins are made, but they do that in a special way. So the protein is the actual person or the soldier that's doing the work. The DNA tells the protein what to do by how it's made, but the DNA never leaves the nucleus in your cells. Certainly in cells that don't have bound membranes, the DNA isn't in a nucleus, but it still doesn't leave the area. So it's still going to use helpers. And so genes are made of DNA, which is a nucleic acid, and there are basically two categories of nucleic acids. There's a deoxyribonucleic acid, DNA, and there's ribonucleic acid, which is RNA. What happens is the DNA is going to give directions for its own replication. And we have, if you look on the right, you have the top image where you have DNA, and it's very simply illustrated as that double helix, so two strands or two bands. And what's going to happen are those bands are going to come apart. That RNA is going to basically be a stand-in for one of the bands. So you have those two ribbons, those blue ribbons of DNA, they are going to pull apart, and the RNA is going to be a stand-in for one of those bands. So it's going to create a, I guess, a mirror image of the strand that it's standing against. It has the capacity to move in and out of the nucleus of the cell. You can see the band, the pink band coming out. It also has the capacity to bring ribosomes, which are organelles that are found in the cytoplasm, to itself and direct the stringing together of amino acids into what we already know is a polypeptide chain. So henceforth, DNA has the ability to create proteins or direct what proteins are made, but never leave the nucleus. So DNA provides the directions for its own replication. That's true. And DNA provides the directions for proteins that are needed by the body. This is done via messenger RNA. So we generally call that lowercase m RNA. And through messenger RNA controls the synthesis of protein, because the messenger RNA can leave the nucleus. You also need an organelle called a ribosome. The ribosome, you can think of it as the mechanism that staples the amino acids together. So brings the amino acids and staples them together. Nucleic acids are polymers called polynucleotides. Each nucleotide consists of a nitrogen base, pentose sugar, and a phosphate group, and I'm sure I have an image of that next. I can point that out. The nucleotide without the phosphate group is called a nucleoside. So we have an image here. This comes right out of your book, so you can see this nucleoside, and the picture in the middle is showing you that is the base plus the sugar. So the nucleoside, you have a 5-ring carbon sugar, a pentose, and a nitrogen base. That's a nucleoside. When we add that phosphate group on, we have a nucleotide. And we string those nucleotides together with a sugar backbone. So the sugar backbone remains the same. Remember, it's deoxyribose in DNA and ribose in RNA, but the nitrogen base changes. The nitrogen base is different. So the backbone is the same, and that's the sugar, the nitrogen base changes based on whether it's DNA or RNA. And there are two families of nitrogen bases, and those are pyrimidines. And so we have our pyrimidines. If you look at the far right, pyrimidines are at the top, and that's cytosine and thymine in DNA and uracil in RNA. Sorry, that one got cut off. Again, this image is in your book. So you have two types of nitrogen bases: pyrimidines, which is a 5-carbon sugar, and then it's a 6-membered ring, and that's the not a 5-carbon sugar. It's a 6-membered ring, and then it's that's cytosine and thymine, and that's in DNA, and uracil in RNA. And then you have purines, which is adenine and guanine, which is a 6-membered ring fused to a 5-membered ring. So it's a much larger molecule because you have 2 rings fused together. And then you have your 2 sugars at the bottom, your deoxyribose and your ribose. Now remember, the nucleotide is where you're adding that phosphate group on, and they are nucleotides are what are linked together to form a polynucleotide. Nucleotides are joined by covalent bonds that form between the hydroxyl group on the 3-carbon of one nucleotide. Remember, when we're saying a 3-carbon, we're numbering these. So you're starting at the farthest to the right, and then numbering clockwise. So you have a 3-carbon on one nucleotide with a 5-carbon on another nucleotide. So with this, we have to go to the image that's it's still on the right, but the furthest to the left on the right, where at the end, if you start at the top, you have the 5 end. So that's the 5-carbon ring, and that one's bonded to the next one by the 3-carbon ring. That becomes very important, particularly when we talk about how this DNA actually goes about replicating itself, that you have this 5 3-carbon ring arrangement, because it always stays this way. They are that sequence of bases with regard to DNA or messenger RNA is unique for each gene. So each gene you have in your body, that sequence is going to be unique to it. So what the sequence of the bases, of the nitrogenous bases. Here is your double helix, which is your DNA molecule with the sugar backbones with the opposite 5-3 direction in parallel. They're actually an anti-parallel, where one side will have a 5 at the top, the other side will have a 3 at the top. You can see that on the far right, you have the 5-3, and that gives you that very, very predictable ladder base. The DNA pair hydrogen bonds form adenine always pairs with thymine, and guanine always pairs with cytosol. In RNA, adenine is going to bond with uracil. In RNA, there is no thymine. And this is what you want to be able to do. The 4 classes, what are they? What a glycoside linkage is, distinguish between a monosaccharide, disaccharide, what saturated, unsaturated fat is, and 4 levels of protein structure, and then information about DNA, given the difference between a nucleotide inside, ribose and deoxyribose, the 5-3 end of a nucleotide. All of this is because we'll build on this material, so you try to have this in your head, and we will pick up on the next chapter.